Hi, everyone. Thanks for tuning in to listen to my talk today. I will be going over some recent work that we've done with respect to bile acids, particularly as it relates to understanding how the host and, in this case, the gut microbiome interact. And I hope to be able to demonstrate to you how important metabolomics has been in helping us to uncover this important role. I'm happy to answer additional questions at the end if time permits. If not, please feel free to send me an email at adp117@psu.edu. First, just like to start off by thanking some important people in my lab. Jingwei Kai, a really talented graduate student who's now a postdoc at Genentech. Yuan Tian, who's an amazing NMR spectroscopist in my lab. Bipin Ramal, really talented graduate student who focuses mostly on bioinformatics. Eric Allman, who's now left to go to AstraZeneca, as well as Bob Nichols, who, again, really talented graduate student who's focused on extensive applications with respect to bioinformatics and chemoinformatics of the microbiome and the metabolome. Lastly, I'd like to thank a longstanding collaboration I have with Frank Gonzalez at the Lab of Metabolism, and also funding sources from NIH, including NIEHS, the National Cancer Institute, and also the PA Department of Health. So what is the metabolome? We think of it quite simplistically as a way to inform us about mechanism in certain metabolic conditions. So in this case, we can use metabolites to help us inform us, or to help inform us about important changes that may be occurring within the host as well as within the microbiome. One of the best ways to kind of picture this or visualize this is with respect to trimethylamine. We know trimethylamine to have a variety of important roles with respect to the microbiome, but in this case, we know that trimethylamine is the common problem associated with fish odor syndrome, and the pathway was worked out through some elegant biochemical assays, as well as some studies of some key mutant chickens. In this case, these are chickens that laid eggs with fishy taint. This was ultimately then worked back to help to identify a mutation in FMO3. Fish odor syndrome was actually also described by Shakespeare, perhaps in The Tempest, in his description of Caliban, who was likely suffering from fish odor syndrome as well. Within the lab, we use a variety of tools and platforms, including triple quads, Orbitrap, and QTOF mass spectrometers. In this case, triple quads for doing targeted profiling of bile acids, including those from rodents and humans. I'll talk a little bit about that data today. Also, Orbitrap mass spectrometers, in particular for doing a lot of unknown identification based on their ability to do MSN, and also some features from Thermo, including AcquireX. We also do a lot of work with NMR, allows us to profile, not a huge number of metabolites, but we can do it in a rather quantitative way. And ultimately, I know this is probably well understood within this group, but we need to have all these platforms because the varied physical chemical properties of metabolites that we're trying to measure, it's just not possible to use one platform to measure all of these currently. Within the lab, we have a lot of different tools that help us to work on this signaling pathway or this communication network between the host and the microbes. Mouse models, whether they're conventional or gnotobiotic, or those that have a defined bacterial population. We use in vitro models and bioreactors that help kind of remove the host from the equation. Sequencing-based approaches, but important to this talk today, a lot of metabolomic tools, including NMR and mass spec, that allow us to really capture this chemical signaling that exists between the host and the microbes. And the important thing is we think about this as the dialogue that exists, and it's a way that these organisms can communicate with us and vice versa. And ultimately, we use a tremendous amount of bioinformatic tools to help us try to look at this in an integrated and perhaps more simplistic view that helps us to really focus on the important pathways that are critical to the exchange of this information between the host and the microbes. So how important are metabolites in understanding the microbiome? We like to think about this in terms of chemical currency. There's a lot of energy being put into the generation of some of these metabolites, or the consumption of some of these metabolites, or the modification of some of these metabolites. This is in some cases, an energetic process. And so that means it's likely to be something that the microbes are doing in a very important and critical fashion, such that they can then modify their environment or make it more hospitable to their overall survival. Some of the classic pathways, the balance between glycolysis and fermentation, particularly as it relates to the balance of NAD and NADH, the production of certain volatile compounds like cresols, generation of short-chain fatty acids through the metabolism of undigestible fiber, metabolism of amino acids like tryptophan to generate indoles, which I'll talk about in a little bit. Perhaps one of the most important ones to today's talk, the generation of secondary bile acids from primary bile acids through the activity of 7-alpha dehydroxylase. And the reason why I bring up just some of these is because we know in the case of some of these bile acids like DCA and LCA, they may be associated with the development of hepatocellular carcinoma, in this case shown here in a mouse liver, but this has also now been associated with the human hepatocellular carcinoma as well. We know that some of these molecules like indoles, as well as some of these bile acids, are important signaling molecules that interact with host receptors, in this case, ligand-activated transcription factors. Indole, for example, is known to bind to the aryl hydrocarbon receptor and activate it. And then also, there's lots of evidence to date to show how important metabolomics has been in identifying key metabolites that are associated with various diseases. Those that I have highlighted here with the smiley faces, like TMA and TMAO, are associated with cardiovascular disease. Changes in different bile acid pools are now linked to things like obesity, diabetes, fatty liver disease, as well as non-alcoholic steatohepatitis. And importantly, it's worth mentioning some of the role that metabolomics has played in identifying new mechanism of action with respect to things like metformin, digoxin, acetaminophen, as well as chemotherapeutic agents like irinotecan, and how important microbes are in the regulation of some of, or I should say, the efficacy of some of these drugs. So the question is, if the microbiome is talking to us and we're trying to talk to it, how are we doing this? So within the host, who's listening? So a lot of our work and some of the work I'll talk about today, centers on the nuclear receptor superfamily, in particular, these ligand-activated transcription factors that fall within the metabolic sensor family of the nuclear receptor superfamily. Today, I'm only going to talk about one of those, the farnesoid X receptor, which we know to be important for a variety of things, including lipid, bile, and glucose homeostasis. So as I mentioned, these are ligand-activated transcription factors, meaning they have to be bound by ligand to exert activity or have their activity repressed. And so in this case, I just show the crystal structure of FXR bound to its coactivator RXR. In this case, being bound to an agonist, in this case, something that's going to turn on its activity for, in this case, is a therapeutic agent to specifically target FXR. So these work primarily through the modification of their activity through ligands. So they exist in a somewhat silent state when bound by co-repressor, meaning that their transcriptional activity is reduced. Apologies for my dog. But then they can be bound to ligand, where they then are activated through the activity of certain coactivator recruitment, as well as a variety of other cofactors, which then can be important for turning on the transcription of key target genes, in this case with FXR, those genes that are important for bile acid metabolism. So metabolomics was adopted, in this case in our lab, to help us to identify some of these key ligands that are important for this process. So those ligands that are known to interact with these receptors, in this case FXR, to activate or turn on its activity. So as I mentioned, FXR, the farnesoid X receptor, is a key regulator of glucose, lipid, and bile acid metabolism. It exists in a variety of tissues, but important for today's talk, it exists within the liver and the small intestine. And a lot of the key signaling molecules are being exchanged through the portal vein, as well as being released from the gallbladder into the small intestine. These key molecules are bile acids themselves. They bind to FXR to either turn on specific activity that indicates that more bile acids need to be made, or furthermore, it turns off key activity that suggests that we have enough bile acids, we don't need to make more. But ultimately, we've been able to identify that some of these key signaling pathways can then also be important for regulating key things related to these pathways like glucose and lipid homeostasis. It's important to point out that we're talking about a tremendous amount of material, so about 12 to 30 grams of bile acids are secreted into our intestine daily, particularly after a meal. And we've begun to appreciate not only how important these are for things like fat solubilization, but also the incredibly diverse and richness of the bile acid pool that suggests that it is much more than just an important detergent, but that it does have key signaling properties. So I will focus on some roles of FXR today with respect to the development of things like steatohepatitis and steatosis, as well as its ability to reduce or ameliorate symptoms associated with obesity, insulin resistance, as well as fatty liver disease. There are some important things to point out. FXR activity in the liver is somewhat distinct from FXR activity in the small intestine. So FXR, when activated in the liver, is thought to help repress or reverse the development of steatosis and non-alcoholic steatohepatitis. Alternatively, if we activate it with, or excuse me, if we inhibit it within the small intestine, we can also see the same exact changes as if we activated it in the liver. Now, this kind of seemingly conflicting approach is still being worked out, but it does suggest that the roles of FXR are important. You know, this is very important to remember, are context dependent. So as I mentioned, we'll begin focusing a little bit on bile acids today. So I just want to share with you a little bit of our bile acid profiling protocol. So this was developed for not only for rodents, but also for humans. It involves a rather standard extraction protocol, whether we're talking about tissues or biofluids or stool or fecal samples. And then these bile acids are profiled, in this case, through a triple quadrupole, in this case, Waters TQS, which we then can do pathway analysis to identify key changes in those bile acid pools. So as of very recently, we were looking at approximately 70, more than 70 different bile acid species. This is now, over the last few months, it's expanded to greater than 100, and it's continuously being expanded based on comparison with public databases. And I just want to really emphasize how critical the public databases are to not only our research, but to everybody else's research. And, fortunately, there's been a lot of effort to continue the funding of these important databases, because it does allow us to do discovery-based work that we otherwise couldn't do in our own lab. So I want to kind of summarize, a lot of different experiments that we conducted over the last few years. And this has all been done largely in mice, although I will end my talk with a discussion of some of this bile acid profiling experiments in humans. Mice treated with really potent antioxidants, antibiotics, looking at mice that are germ-free, or looking at mice that are monocolonized, meaning they're only germ-free mice that are colonized with a specific bacteria. We really were able to identify some key changes in different bile acid pools, whether we're looking at those that were obtained from the feces or stool, and comparing those bile acid species to those that were found in the small intestine. And what we found was, of these treatments, or when using all of these treatments individually, we found that we could see an enrichment in unconjugated bile acids within the systemic circulation, or excuse me, within the small intestine. However, those unconjugated bile acids were freely excreted in the rodent stool. However, what we found was that those bile acids, when we looked at those that were present in the small intestine, those conjugated bile acids were enriched following these treatments. And the question was, why is this? So again, just to kind of reiterate this, fecal bile acids, we found unconjugated bile acids were depleted in this bile acid rich sample. If we looked at the small intestine, in this case, predominantly the ileum, we found that conjugated bile acids were enriched following either one of these treatments. So what are the things that were driving this? Ultimately, we were able to identify this key pathway that's present in a variety of different bacterial species. So that is the metabolism of bile acids through this key enzyme known as bile salt hydrolase. Bile salt hydrolase is common to many commensal bacteria. It helps to provide bacteria specific bile acid tolerance. So otherwise, these bile acids are quite toxic. We've published a few papers to suggest that this deconjugation pathway, or the removal of this case of taurine from taurine beta-murocholic acid, is perhaps a key scavenging event that provides some energy for the organism as well. And most importantly, these commensals need this bile salt hydrolase for gastrointestinal persistence, meaning they need this to be able to stay colonized within the niche of the small intestine. And through a variety of different assays, we ultimately were able to find that these bile acids, in particular, some of these conjugated bile acids, namely taurine beta-murocholic acid, is a potent FXR antagonist. So this means that this bile acid is key in downregulating or reducing the activity of FXR. And what I'm showing here is just a key reporter assay where we treat with either TCA, in this case taurocolic acid, which is an FXR agonist. It turns on its activity. Whereas if we treat with T-beta MCA, you can see that that activity is reduced. Now, as I mentioned, BS activity is really important for intestinal colonization and GI persistence. In this case, what we did was we colonized mice with different types of bile salt hydrolase active bacteria. In this case, L. delbrueckii 11842 that does not have BSH. Compare that to L. johnsonii or L. acidophilus. And note that if we colonize these mice with these different bile acid containing bacteria, those that lack BSH activity do not persist within the animal following, in this case, with L. delbrueckii, about 3 weeks. Those that have BSH activity colonize and remain throughout the study timeframe. And so what we were able to identify is that this was a key pathway by which bile acid pools are being regulated within the small intestine of animals. And I'll also show some data about this, how this exists in humans. So we can do this a number of ways. We can either specifically deplete this bacterial species through antibiotics. We can also inhibit its activity through certain inhibitors, in this case, caffeic acid phenylethyl ester, known as CAPE. So the reduction of this activity increases the level of this T beta MCA, which is then likely to repress the activity of FXR. We've also generated bile salt hydrolase resistant bile acids, in this case by swapping out taurine, in this case with glycine. So we kind of have a pseudo-human rodent bile acid, which unlike T beta MCA is resistant to BSH activity. So, and furthermore, is important that we can use this as a therapeutic agent to reduce the activity of FXR in the small intestine. And so this just summarizes some of the things I've just mentioned. So we know that T beta MCA can be metabolized by BSH to generate uracolic acid. We can inhibit this activity through specific BSH inhibitors. When we do so, this enriches T beta MCA, which we know to inhibit FXR. We can also deplete the bacteria through either antibiotics or specific potent antioxidants, known as, in this case, with Tempol, which also can inhibit FXR. And furthermore, we can generate certain bacteria that are, or enrich for populations that are bacterial populations that lack BSH activity, which also can lead to the enrichment of T beta MCA, which can also inhibit FXR. And the reason why this is so important, and this is summarized as a tremendous amount of work that we've published throughout the years through the journals listed here, including Nature Communications, JCI, Diabetes, and that's kind of described within this pathway here. A lot of this has to do with changes in bile acid pools themselves, but ultimately we think that this is a pathway by which FXR regulates or is a key regulator of important things like these certain toxic lipids, ceramides, that we think are likely to be important for the production of things like non-alcoholic fatty liver disease and steatosis. So if we turn on FXR in the small intestine, this then promotes the development of fatty liver disease. We can block that through specific bile acids, including some of our synthetic bile acids like glycine beta-uracolic acid. And in doing so, we can either prevent or reverse the development of fatty liver disease, specifically by targeting FXR in the small intestine. And so there's a lot of other work related to this that I don't have time to go into. Has a lot to do with the beiging of adipocytes, as well as some important changes that occur in the liver as well. And I'd be happy to talk about that afterwards. So what does this have to do with the human population? So I just want to end here with my last few slides talking about the translational connection that we've made from some of our rodent studies with respect to an important human medication, in this case, metformin. So metformin is one of the major therapeutics for type 2 diabetes. It's been in use in the world in around 1950s. It's been around in the world since the 1950s, but it's only been approved for use in the US since 1995. It was originally identified in a search for a number of different compounds, but some of those derivatives were abandoned due to toxicity. Metformin itself was rediscovered during a search for antimalarials and repurposed by a French physician for type 2 diabetes. The interesting thing is its overall mechanism of action is still not fully worked out, but there is some hint that the microbes themselves, the microbiome is key to its activity. And so what I'm showing here is a microbiome sequencing-based analysis where individuals that are type 2 diabetic that were naive to metformin, we profiled their stool samples before and after metformin treatment, and then look to see which bacterial species changed after the treatment. And one of the key ones we found were changes in B. fragilis, which importantly we know to have important bile salt hydrolase activity. So key to the the observation that I just mentioned in the rodent models. And so using that same bile acid profiling tool that I described earlier, we profiled a variety of different bile acid species. And one of the key ones that we found was an enrichment of certain bile acids and conjugated bile acids, in this case, GUDCA as well as TUDCA, which we were able to identify based on the reduction of this bile salt hydrolase activity following treatment with metformin. So this suggested to us that metformin was specifically depleting, or potentially, we haven't worked this out yet, killing off key bacteria that have bile salt hydrolase activity. So if we do a simple culture experiment where we grow wild type B. fragilis or mutant B. fragilis that lacks BSH in the presence of bile acids, we can see that those that have BSH will deconjugate bile acids, and they will then fall out of solution and precipitate around the colony. Those mutants do not have this activity, so they cannot precipitate, or they cannot deconjugate, and these bile acids then do not precipitate. So this is really critical observation that as BSH activity falls, we found this to be indirectly proportional to FXR repression. And one of the key observations that we made was that some of the enrichment of these bile acids were key to understanding the depletion or the inactivation of FXR in some of these pathways. And so here I'm just showing an important co-activator recruitment assay where we look at an FXR agonist, CDCA, with respect to differences in some of the antagonists that we identified, GUDCA and TUDCA. And so in summary, this summarizes a tremendous amount of work. What we think is happening is that metformin is depleting or killing off specific things like B. fragilis. This reduces their BSH activity. This alters this bile acid pool, enriches for FXR antagonists. And much like what I showed in that model in the rodents, the enrichment of these bile acids antagonizes FXR, which then may be important for the reduction or the amelioration of things like fatty liver disease in humans. So just to end with some important remarks, we have been able to really capture a lot of these key changes that are occurring within the microbial community structure. We're able to monitor their metabolic activity through metabolomics and ultimately, hopefully, link these changes to certain health outcomes. Some of the key things that are critical and where metabolomics has been most informative is in understanding how these different perturbations are driving changes in microbiome metabolism, including things related to the host metabolism, genotype, immune system. And more importantly, we're beginning to appreciate how xenobiotics may also impact microbial metabolism as well, to ultimately have some impact on human health and disease. So lastly, I just want to thank some key people within the lab at Penn State, talented postdocs who I've already mentioned, other key collaborators, including Costas, Chantu and Demont, who did a lot of the chemical synthesis, Gary Perdue and his group, who did a lot of work with AHR, some new work that we've been doing with respect to mass spec imaging with Nick Winograd and his talented research professor, Teresa Hu, as well as key people within Frank's group, including Chang Tao Jiang as well. So thanks very much. I appreciate your attention and be happy to take any questions following the end of this session. Thank you.